Okay, I think we can start. Welcome, everyone. I am Sera Monti. I'm a rheumatologist from Italy. It is my great pleasure to introduce the first of a series of webinars. These have been organized in collaboration with EUVAS, the European Vasculitis Society, and it will be a Vasculitis Awareness Month. We'll have a webinar today and then 3 more every Tuesday of May. So you're all very welcome to participate and subscribe to the following ones as well. Today, the webinar will be on monogenic vasculitis. This is a great topic, and I'll hand it over to the chair of the session, Ahmet. Thank you very much for joining.
Well, thank you very much, Sarah, for the nice introduction. Well, you're very welcome to the first of the series of ERN-RITA webinar series organized in collaboration with European Vasculitis Society. So can I have I hope you can see them now. So this series will include some interesting topics, which would be quite important for the Vasculitis Society and also for the European Reference Network in association with the rare inflammatory disorders. And today's topic would be monogenic vasculitis. As you know, vasculitis is inflammation of blood vessels, and we have been using the nomenclature developed at the 2012 Chapel Hill Consensus Conference, which usually aims to define different vasculitic conditions depending on the types or sizes of blood vessels. And the major groups include large vessel vasculitis, medium vessel vasculitis, and also small vessel vasculitis. And there's also a variable vessel vasculitis type. But if you look at the remaining parts, there is also a group of vasculitis which have some probable etiologies. Among them, we previously recognized different infections or drugs or malignancies associated with vasculitic manifestations. But today, we will focus on monogenic disorders, which have vasculitic manifestations as their main clinical features. So the first speaker, we have 2 outstanding speakers today. The first speaker will be Dr. Seza Özen. She is the president-elect of the PRESS, Pediatric Rheumatology European Society, and a professor and head of the Department of Pediatric Rheumatology at Hacettepe University Faculty of Medicine in Ankara, Turkey. Her research focuses on vasculitis, autoinflammation, and chest disease. She graduated with her medical degree from Hacettepe University. She thereafter completed a residency in the university's Department of Pediatrics and also a postdoctoral fellowship at Stanford University Medical Center in California. Since 1992, Professor Özen has served in Hacettepe University Faculty of Medicine as an assistant, associate, and now full professor. She is a steering committee member of the Eurofever project. She also has received several grants as principal investigator from national and international projects. She has received 2018 Medical Prize TWAS and various national prizes. In addition to authoring chapters in 11 main textbooks, including Nelson Textbook of Pediatrics, Dr. Özen also published over 600 journal articles with over 25,000 citations. She has given many invited lectures and conferences and grand rounds in meetings and universities over the world, and that would be just another one within this webinar. So it would be a great pleasure to listen to you, Seza.
Thank you so much, Ahmet, for that kind introduction. Can I have my slides, please? Right. And while I'm waiting for my slides, yes. Can you see my slides?
Yes, yes, we can. Yes. And maybe just before starting, I have to warn our audience about their questions. Whenever they have a question, they can easily send these questions to us using the chat platform. So we would be more than happy to answer them at the end of each talk.
Thank you, Ahmet, for that kind introduction. And I must say it is a real honor and challenge for me to speak at the same session with Dr. Dan Kastner. Dan Kastner has opened a new chapter, a great new chapter, and brought a new concept, not just in rheumatology, but in medicine as well. So I try to follow his steps to flourish the field of autoinflammation, and I will do my best today in this session. So I will talk about monogenic vasculitis. These are my disclosures. And I will carry on from where Ahmet left it. So in this Chapel-Hill classification criteria, there were the primary vasculitides and there were the secondary vasculitides. And the monogenic vasculitides we will talk about today pretty much fit the secondary vasculitides with a known probable etiology. There may be a number of ways we can classify these monogenic causes of vasculitis. There are some that are associated with a known defined immunodeficiency. We all have patients with, for example, BRCA1 deficiency. We have had one that resembled an ANCA-associated vasculitis, or vasculitis, systemic vasculitis can be associated with TAP2 deficiency. But then there are those where there is an autoinflammatory disease, and vasculitis or vasculopathy is a feature of the disease per se. One of these is SAVI, an interferonopathy. Another one is a haploinsufficiency of A20, and the other NF-kappa-biopathies, if I want to call them. There is then deficiency of ADA2, VEXAS, and others, for example, the lion kinase that was associated with small vessel vasculitis. Now, today we decided to cover the two most common ones. So I will talk about the deficiency of ADA2, and I will refer to it as ZETA2, and Dr. Kastner will talk about VEXAS. But I have backup slides about the others if there are questions, or I'll be happy to share them via email. So this was a figure that my co-speaker, Dr. Kastner, has suggested, and I show it in all my lectures, I think, especially to the students, because I think we now have a nice understanding of how, where common variants were implicated in our common diseases, we do have sometimes very rare, very bad mutations that can present with the same future of that multifactorial polygenic disease. We knew that, for example, C1q deficiency was an example for lupus, but with these rare vasculitides, we know that they are also representations of rare mutations with causing mimicking our common diseases. And ADA2 deficiency in this sense was a game changer because it's a total mimic of polyarteritis nodosa. It's a vasculopathy with autoinflammatory features, and actually this livedoid racemosa is probably the most common feature. I've seen it in all of my patients, and they have a very frequent strokes, which again, distinguish them from other some autoinflammatory diseases. They are due to the loss of function mutations in the gene coding for adenosine deaminase, and we have learned about them only up in 2015. And when we went back in 2015 and looked at our patients, we found that some of them whom we thought had polyarteritis nodosa actually had ZETA2, and they needed to be treated differently. So they do have some atypical features that might help you to distinguish from polyarteritis nodosa. They have more tissue loss. They have the recurrent strokes. They have recurrent fever as well, and sometimes they have been mistaken for the other autoinflammatory diseases. And they are refractory, usually, to conventional immunosuppressive treatments that we use for polyarteritis nodosa. We know a bit about the pathogenesis. We know that ADA2 is very different from ADA1. You know that ADA1 deficiency causes a very severe immune deficiency, whereas the ADA2 deficiency acts first by changing the macrophage polarization and then also altering the endothelial cell integrity, causing damage, and with the dysregulation of netosis through the chronic neutrophil activation. So this was a study again from the NIH where they have shown that indeed netosis is a very important feature in the pathogenesis, and the TNF-alpha that we see in these patients was probably produced as such. And as I will come back, anti-TNF is the main treatment for these patients. So they have shown that the more you put adenosine, the more nets you get by the neutrophils, and less nets when ADA2 is added. So when we come back to the clinics, the main phenotypes in the literature are, for one, the stroke and vasculopathy, the pan-like phenotype, but then they can also present with immunodeficiency and hematological features. And I thank Ezgi for sharing this figure with me, because it does show how multifaceted the two can be. Now, recently, a group of experts, we came together and we tried to develop some management recommendations and evaluation recommendations for DADA2. And in this paper, we reviewed that there were mainly 4 presentations of this disease, although there might be sometimes mixed features: pre-symptomatic, inflammatory, endovasculitic, hematologic, and immunodeficient. And the inflammatory endovasculitic features would be with patients with recurrent fever episodes, because fever is the main manifestation of inflammation, high inflammatory markers, manifestations of vasculitis, including cutaneous vasculitis, and very much so livedo racemosa, ischemic stroke, sometimes intracranial hemorrhage, sometimes neurological features, and tissue loss. And then we have patients who might present with mainly the hematologic features, and they sometimes mimic diamond blackened anemia. They may present with variable anemia, neutropenia, thrombocytopenia, and even autoimmune hemolytic anemia. And they may also represent with lymphoproliferation resembling autoimmune lymphoproliferative disease, and cases of lymphoma have also been reported. The 4th presentation is immunodeficiency. They might present with hypogammaglobulinemia, lymphopenia, or recurring opportunistic infections. Now, of course, the inflammatory and vasculitic patients can have some hematologic features, and they do often have low IgG levels, or the immunodeficient patient may also have some inflammatory features, or the hematological presentation might also have some stroke or other vasculitic features as well. So they can present in multiple ways. So if they can intersect, but so they either present with vasculopathy and inflammation, immunodeficiency, or hematological features. The vasculopathy and inflammation can present itself with constitutional symptoms, with very severe GI disease, with other organ involvement, including nervous system involvement, arthralgia, arthritis, myalgia, digital ischemia, digital tissue loss, renal infarcts, renal aneurysms, very much mimicking polyarteritis nodosa. And in the cardiopulmonary system, myocarditis, cardiomyopathy, pleuritis, pericarditis. So really, there's a wide range of manifestations. This was our experience in our single center in over 25 patients. The age of onset was low. They might present in childhood. They do have a necrotizing vasculitis in medium and small size arteries when you biopsy them. And actually, our pathologist, who's quite experienced, told me that there was no way she could differentiate between the biopsy of a PAM patient versus a DADA2 patient. And again, their aneurysms very much look like the ones we see in polyarteritis nodosa. The system involvements, as I said, dermatological features are present in almost all, GI findings, neurological findings, renal findings, ophthalmologic findings, especially strabismus, are again very common. Half of our patients have parental consanguinity, and half of them have, further some evidence. Arterial aneurysms were present in more than 2/3, and actually more than 1/4 had some low IgG levels or other immunologic findings, and some had hematologic findings. And especially the adult patients who were diagnosed late when the disease was first defined were lost. So how do we diagnose them? We diagnose them either or by the ETA2 enzyme level testing and/or the mutation testing for the ETA2 gene. And Pui Li's work from Boston has nicely shown that actually in patients with bone marrow failure, there is almost no ETA2 activity, whereas in the patients who present with vasculitic inflammatory features, you might expect some. So again, in the same paper that I mentioned, the consensus paper, we reviewed some, we suggested some statements for how to diagnose these patients. And as I said, we suggest the ETA2 enzymatic testing. We suggest the, if you can't do that, mutation analysis either is accepted if they are pathogenic. And we also say that individuals with clinical features of the two, but non-confirmatory genotypes should undergo further evaluation with other two enzyme activity and or additional genetic tests. So if you strongly think about this disease, you should insist on your analysis. This was our results when we looked at our results. The two patients with hematological presentation and vasculitis-like presentation, PAN-like features, had very, very low ETA2 enzyme levels, whereas healthy controls in polyarteritis nodosa patients, the true polyarteritis nodosa patients, had normal ones. And the heterozygotes, the mothers and fathers, had levels in between, but they had no symptoms. And this was our mutational screening. It's not a very big gene, so it's not so hard to do the mutation analysis. So in the same paper, we also suggested that you should screen siblings. Now, this is, of course, a problem when you're screening a sibling who has been just born, a newborn, because it's hard to give the treatment for a newborn, but this is something that we have to consider. We have also suggested that screening should be considered for all individuals, especially with pediatric onset polyarteritis nodosa. And that's what we do now. Whenever we diagnose a child with polyarteritis nodosa, we also do a genetic screening for other two deficiency. We have also suggested to do the screening for Sneddon syndrome and maybe levetirazonosa associated with inflammation, or really unexplained autoinflammatory features that suggested the two. We have also suggested that screening should be considered for individuals with cryptogenic ischemic stroke, especially those starting early in childhood, adolescent, or early adulthood, and for individuals with unexplained brain hemorrhage, because these patients might present with ischemic strokes or hemorrhagic strokes, or even spinal cord strokes. So again, there is, although the level of evidence is not high, we have suggested screening for these patients. And also screening should be considered for unexplained bone marrow failures and unexplained immunodeficiencies with low Ig levels. Again, we carry on these statements with what we suggest to do when you suspect a patient. Of course, the physical examination is important. The complete blood count is very important, as I will come back to in the next slide. The good phase reaction is important. And of course, the organ functions should be evaluated as necessary. We always check for serum immunoglobulin levels because in more than 1/4 they will be low. You might need to do angiographies if there is stroke. And additional testing is really based on what you see in the futures and the organ-specific manifestations. It is important that the care of the DATU will require a multidisciplinary team of healthcare. This is important because it is such a a multi-system disease that requires many specialties. Patients with controlled disease should be followed by experienced clinicians at least every 3 to 6 months. So I think the complete blood count is important because when we compared our the 2 patients with our polyarteritis nodosa patients in our center, we saw that something that was quite different. They both had high CRPs and everything, and their futures looked similar, although there was more stroke in the 2 group. One difference in the simple complete blood count was that we didn't see the typical thrombocytosis we see in polyarteritis nodosa patients. So the thrombocyte counts were either normal or even on the low side. So we think this is helpful in also for your suspicions. As for the management and treatment, so the treatment of the 2 for the time being is anti-TNF, and treatment for anti-TNF is lifelong, unfortunately, in symptomatic patients with inflammatory vasculitic phenotypes. Screening of TB is required, especially in countries where TB is still prevalent or exists. And TNF inhibitors should be considered in patients with overlapping features of vasculitis and immunological or hematological features as well, because it will help the vasculitic and inflammatory features. But the real treatment for hematological involvement is allogeneic stem cell transplantation, unfortunately, because you cannot correct the cytopenia with anti-TNF inhibitors. Again, it's important that antiaggregants, anticoagulants, and antithrombotic therapy is not recommended in the 2 because they have a high risk of having hemorrhagic strokes. Acute management, you can use steroids, but the lifelong treatment is anti-TNF, TNF inhibitors, and you might need immunoglobulin replacements for patients with really low IgG levels. And TNF inhibition does work. Again, my co-chair's group has shown that TNF inhibition does help to improve the vascular integrity, helps the macrophage polarization, and they have also shown that it decreases the interferon signature. Actually, we all know that the interferon signature is increased in ATA2 deficiency, and there are thoughts of maybe grouping it among the autoinflammatory interferonopathies. So the current treatment, as I said, is TNF inhibitors. We know it works in inflammation and vasculitis. For the hematological and maybe severe immunological features, pancytopenia, you would need allogeneic hematopoietic stem cell transplantation, unfortunately, because you can't cure it with TNF inhibitor. But there are treatments in the pipeline that we are looking forward to. There's a group in Italy that is working on gene therapy, and also a group in UK. And there's a group in the US working on enzyme replacement. So we are very much looking forward for these because this is a lifelong disease, and they will need lifelong treatment. And it's very problematic to treat, for example, a newborn. I'm saying this because I know UK is starting a screening program now that will also include checking for the 2 deficiency. And I don't know what they will do with the newborns with the 2. So my concluding remarks are, you need to consider the 2 in any patient with unexplained inflammation, actually in any organ, especially if they have high CRP and they have the liver resevulsa. Many palm lung patients in our region present with pan-like features and not so much with pancytopenia and immunodeficiency. And we don't know if that's because of the high frequency of G47R in this region. Of course, we need collaborations for final answers. So I will end there, showing my group here, and we do hope to become an ERN member country ourselves. So thank you, and I welcome questions. Thank you.
Okay, thank you very much, Seza. That was a wonderful talk. We have some questions, but we may have all of them at the end after finishing Dan's talks as well. So I would be more than happy to introduce our second speaker, who is, again, as I mentioned, an outstanding figure in the area of autoinflammation and also genetic basis of several other conditions. Can I share my slides?
I have stopped sharing mine, right?
Yeah, I think
Okay.
it's okay now.
Okay.
I will start by sharing Dan Kastner. Dan Kastner obtained his undergrad degree with summa cum laude in philosophy from Princeton University in 1973 and a PhD and MD from Baylor College of Medicine by 1982. After completing internal medicine residency and chief residency at Baylor, Dan moved to the NIH in 1985. He served as scientific director of the Division of Intramural Research of the National Human Genome Research Institute from 2011 to 2021, and he continues to maintain a busy lab in the medical genetics branch. Throughout his career at the NIH, Dan's research has focused on using genetic and genomic strategies to understand inherited disorders of inflammation, often stimulated by patients with relatively rare disorders seen at the NIH Clinical Center Hospital. This work has provided detailed molecular explanations for these illnesses, which includes the monogenic vasculitis, has provided the conceptual basis for the highly effective targeted therapies, and has informed our understanding of more common illnesses. Dan's group also proposed the now widely accepted overarching concept of autoinflammatory disease to denote disorders of the evolutionary ancient innate branch of the human immune system. Dr. Kastner has won a number of awards and honors, including election to the National Academy of Sciences in 2010 and to the National Academy of Medicine in 2012, recognition as Federal Employee of the Year in 2018, and the Ross Prize in Molecular Medicine in 2019, and lastly, the Crawford Prize in polyarthritis in 2023. So it would be a great pleasure to listen to you, Dan, about VEXAS.
All right. Well, thank you so much on that for that really kind introduction. It's a real honor being here today, having an opportunity to speak with you all, and it's also an honor to follow Dr. Özen, who, although she's very modest, is actually a major figure in her own right. So in any case, I'm going to fiddle around here with my screen in order perhaps to get it so that you'll be able to, let's see, I've got to go back here. This, I hope, will not make you all dizzy. But in any case, let's get started. So we're going to sort of be going now to the opposite end of the age spectrum in terms of thinking about the presentation of various forms of vasculitis. Dr. Özen has beautifully summarized the story with the deficiency of ADA2, which oftentimes presents very early in life. But now we're going to be talking about a different disease, VEXAS syndrome, which I've designated here in the title as being the next frontier in monogenic vasculitis. It's a disorder that, at least so far, we have only been seeing in individuals, for the most part, males, that are middle-aged or older. Maybe just to get underway, I don't have a disclosure slide, but in fact, I don't have anything to disclose to you in terms of financial relationships. But this talk is going to focus on the question of how do middle-aged and elderly people develop monogenic vasculitis. This was a question that was vexing, if you will. A former fellow in my lab, he was a fellow at the time that he was first thinking about this, David Beck. And he was working along with some other folks in the intramural program of the NIH, Peter Grayson, Marcy Ferrata, Keith Sikora, and Amanda Umbrello. And David was a very impatient guy at this time, and he felt that we were just dragging our feet, where we were going too slow in terms of finding genes that underlie various inflammatory and auto-inflammatory disorders. And he thought that perhaps rather than using a phenotype first approach, which is the classic approach for going after disease genes. Phenotype first means that you take a bunch of individuals who have similar clinical presentations, clinical phenotypes. You try to group them together. You do some sort of genomic analysis on them, whole exome sequencing, whole genome sequencing, and then you look for something in common amongst those individuals that might define a new disease. David, on the other hand, wanted to take the opposite approach, genotype first. And so he did it in a really big way. And so what he did was, rather than just taking one gene or a couple of genes and seeing whether or not he could find a bunch of people that have mutations in that gene or a couple of genes, instead, he made a list of all of the genes in the human genome that he could think of that have something to do with ubiquitination. Now, for those of you who have forgotten your biochemistry, ubiquitination is a post-translational modification of proteins that actually regulates a lot of the roles of proteins in inflammatory responses and in signaling, immune signaling. And so ubiquitination, we know, is a very important process regulating the immune system, both the adaptive and the innate immune system. So there are 841 genes that he could think up that have something to do with ubiquitination. And then he made a list of all of our undiagnosed cases from our autoinflammatory cohort, a list of 1,477 of them for which we had exome or genome data. And then he also convinced Dr. Bill Gall, the head of the Undiagnosed Diseases program, to share with us 1,083 exomes or genomes from the Undiagnosed Diseases cohort. He was looking for genes that are very intolerant of variation, a PLI of greater than 0.9, and not present in this big database. The variants wouldn't be present in this big database called NOMAD, but would be shared amongst the cases. And from this Herculean undertaking that David Beck did, he found 3 middle-aged men who appeared to be heterozygous for variants at residue 41 of this gene, UBA1. Now, UBA1 is a master regulator of ubiquitination, so you could say, wow, he hit the jackpot with that. But it's so important for ubiquitination that you might actually think that, well, that's impossible, that it's impossible to live if you have substantial mutations in that gene. There was another fly in the ointment, and that was that these patients that appeared to be heterozygous for these variants in UBA1, they were middle-aged men, and UBA1 is encoded on the X chromosome. So most men only have one X chromosome, as you all know, and so that became somewhat of a puzzle. How could it be that if men have only one X chromosome, how could these guys be heterozygous for a variant in an X chromosome gene? So in any case, this was a real mystery. One possible explanation would be that it was just a sequencing error. So David ruled that out. A second possibility could be that in fact these guys were aneuploid, that they had an extra X chromosome. David ruled that out. And so the third possibility could be that in fact it was a somatic mutation in these three individuals. In other words, that the mutation arose in some subset of cells in these three middle-aged men. And so the mutation would then be seen in the DNA from those cells, but the normal version of the gene would still be seen in the rest of the cells of the body. So that rather than all of the cells having both versions of the gene, some of the cells in the body would have one version, others of the cells in the body would have the other version, and when you mix the DNA together, it looks like the person's heterozygous. So in any case, that's what David was looking for, and in fact, that's what he found. So looking at this slide, you can see that in the peripheral blood, there are both versions of the gene, both the wild type and the mutant. In the hematopoietic stem cells, also both versions of the gene. But if you look separately at the myeloid cells, you can see a predominance of the mutant form of the gene. And if you look at the lymphocytes, you see a predominance of the wild type form of the gene. If you look at variant allele fraction in different subsets of cells, if you look in fibroblasts, the variant allele fraction, in other words, the mutant fraction, is essentially 0. If you look in the T cells and the B cells, it is essentially 0. But if you look in the myeloid cells, you see that the variant allele fraction can be as high as 80% in some subsets of the cells in some individuals. So in fact, that was the explanation. Now, given the fact that the mutations are present in the bone marrow, we went to our friendly hematopathologist, Dr. Kathy Calvo, and she pointed out to us that in fact in the myeloid and erythroid precursor cells in the bone marrow, that in fact there are these funny looking vacuoles. She didn't remember at first where she had seen these vacuoles before, but ultimately she remembered that actually she had seen them in a couple of our patients from a few years earlier. And one thing led to another, and before you knew it, we had a set of 25 patients, all of whom were middle-aged men, all of whom had somatic mutations in this UBA1 gene, and at least for all of them that we looked, had vacuoles in the bone marrow. Now, interestingly, these 25 patients had carried a whole bunch of different clinical diagnoses before we figured out that they all had UBA1 mutations. And so you can see the list on this slide of the previous diagnoses of these patients: relapsing polychondritis, Sweet syndrome, myelodysplastic syndrome, MGUS, polyarteritis nodosa, and giant cell arteritis. You can see that the percentages actually add up to more than 100% because a lot of these guys had actually carried more than one diagnosis in their diagnostic odyssey, if you will. But in the end, they all had mutations in UBA1. Just looking at what the clinical features of these 25 individuals were was very illustrative. Again, all males. There are a few patients we now know with mutations in UBA1 who are female, but most of the patients are male. Age of onset over the age of 45. 40% of these patients actually had died by the time that we figured this out. So it's a severe disease. Fever, a predominant manifestation. Skin involvement, such as Sweet syndrome, also a very frequent involvement. Pulmonary infiltrates in almost 3/4 of these individuals. Chondritis of the ear and nose. Venous thromboembolus in about half of the patients. Many of the patients with macrocytic anemia, and from all that we looked, vacuoles in the bone marrow. So in any case, we have these middle-aged elderly men who have somatic UBA1 mutations in the myeloid lineage. We thought that it was time to come up with a new disease name that would basically cover this phenomenon, and we came up with the name VEXAS. And VEXAS is an acronym for vacuoles. Of course, you know where the vacuoles are seen in the bone marrow. E1, ubiquitin activating enzyme, that's what UBA1 is. X-linked autoinflammatory somatic syndrome. So it's at least an acronym that can help you remember some of the clinical features of this disease, which are illustrated on this slide. Here you see the pulmonary infiltrates on the scan. If one looks histologically, one can see that it's a neutrophilic alveolitis. These patients have a medium vessel vasculitis. They can have chondritis. They have the vacuoles in the bone marrow. If you look at the neutrophils, you know, you'd say, well, since this is a neutrophil disease, so what do the neutrophils look like? And since there's a lot of them in the peripheral blood with the mutation, is this something that's like a malignancy? And if you look at the neutrophils in these patients, and I guess I should turn on my laser pointer here. If you look at the neutrophils, you can see that they look horrible. This is a Vexus neutrophil, and it looks like it's about ready to die. And in fact, if you remove the neutrophils from these patients and put them in culture, that's exactly what happens, is that they do die. But if you look at just their gene expression profile, if you remove the neutrophils and quickly isolate the RNA, what one can see is that, in fact, there's a very prominent inflammatory gene expression profile, which is not present in the lymphocytes of these patients. So molecularly, what's going on with these patients who have Vexus that have the mutations at methionine 41? Well, there are two start sites, actually, for the UBA1 protein. One of them is at methionine 1. That leads to a longer form of the protein, a higher molecular weight form of the protein that's seen predominantly in the nucleus. That's called UBA1A. And then there's a shorter form of the protein that starts at position 41 that is found in the cytoplasm, and that's called UBA1B. So you see that in the CD3 positive cells, so CD3 positive, those are going to be lymphocytes, and you see that the control individuals and the patients both have a band, a higher molecular weight band for UBA1A, the nuclear form of the protein, and a lower molecular weight band for UBA1B, which is the cytoplasmic form of the protein. So basically, it's normal in the lymphocytes. But if you look at the myeloid cells, you see the controls have a UBA1A band, as do the patients. That's the nuclear form. It's fine. But the cytoplasmic form in the controls, it's there where it should be. But UBA1B in the myeloid cells of the patients is not there. And instead, you get this lower molecular weight band called UBA1C. And you might just think that, well, it probably starts at the next methionine down in the transcript. We wanted to test that hypothesis, and so we did a transfection experiment. We looked at wild type UBA1, and basically you get the A band and the B band. If you look at the mutant version of UBA1 with substitutions at position 41, either valine, threonine, or leucine, what you see is that you lose the UBA1B band, but you get the UBA1C band. Now, the next methionine down in the transcript is at position 67. So if you mutagenize that position 67, then you should lose the UBA1C band, and voila, you see that, in fact, the UBA1C band goes away when you mutagenize position 67 to an alanine. What's more, if you look just to see what's going on with regard to the function of the UBA1 protein, what the UBA1 protein does is that it gets ubiquitinated, actually, and then it transfers that ubiquitin chain to another molecule in a chain of molecules that are involved in the ubiquitination process. Well, if you look at UBA1B, you can see that, in fact, the low molecular weight, which is the unmodified form of UBA1B, there's very little of it. Most of it does get ubiquitinated. Whereas UBA1C, you have mostly low molecular weight. It's unubiquitinated. Very little of it is ubiquitinated. In other words, basically, these mutations are loss of function mutations in this gene. So, in any case, that's at least a little bit of what's going on with regard to this new disease, Vexus. And we published this, if you can believe it, in the New Year's Eve edition of the New England Journal of Medicine at the very end of 2020. Well, in any case, what has gone on since then? Well, for one thing, Marcy Ferrata, a colleague at the NIH, whose picture I showed you a few slides ago, has a very large cohort of patients with relapsing polychondritis, 92 such patients with relapsing polychondritis. And just to give you an idea, what percentage of patients with relapsing polychondritis have UBA1 mutations? The answer to that question, at least in that cohort, was 7 out of the 92. Those 7 were all males. They were over the age of 45 at the age of onset, and their clinical condition included fever, ear chondritis, skin involvement, such as Sweet syndrome, deep venous thrombosis, and pulmonary infiltrates. No chondritis in these patients of the airways or no costochondritis. Macrocytic anemia was seen almost in all of them, with thrombocytopenia. And so if one has a patient with relapsing polychondritis and you want to think about, without actually doing the sequencing, whether the patient has a UBA1 mutation, clues would be male gender, an MCV greater than 100, and a platelet count less than 200. And that actually, in this cohort of 92 patients, was pretty good in terms of determining who had a UBA1 mutation. If one thinks about the survival of patients with VEXAS, I have already told you that this is a pretty bad disease, where 40% of that initial cohort of 25 patients had already passed away by the time that we figured it out. And you can see actually on the left-hand side that roughly 50% survival after 10 years overall amongst the larger cohorts of patients. And if one looks at the patients who have the valine mutation, which is shown in red here, you can see that they have a much worse prognosis. Now, trying to explain that, if you look in the lower panels there, if you look at the lower panels there, what you can see is that the valine mutation is associated with less UBA1B. In other words, it's a variant where you get even less UBA1 activity in the cytoplasm. So it really is a loss of function kind of thing. If you think about other genotype phenotype issues with these patients, this is another paper from Marcy Ferrata, and one can see that the valine mutation is associated with an increased risk of death, as is the dependence on transfusions, whereas actually ear chondritis is associated with a slightly reduced risk of death. Here are some other genotype-phenotype relationships I won't go through just for lack of time. Here's, though, another paper that's really a major paper in the field of VEXAS. This is a paper that appeared in the Journal of the American Medical Association back in early 2023. David Beck was the first author of it, and it was a collaboration with the Geisinger Clinic. Now, the Geisinger Clinic is a clinic system that is in northern Pennsylvania. It's a set of a bunch of different clinics spread out in small communities in northeastern Pennsylvania. And they have about 163,000 patients who have undergone sequencing, either exome or genome sequencing. And in all 163,000, they then looked to see whether or not they had UBA1 mutations. And they found that 12 out of the 163,000 individuals, in fact, had a UBA1 mutation, leading to an estimated prevalence of about 1 in 14,000. About 1 in 4,000 men over the age of 50 were found to have UBA1 mutations. So if you think about that across the world, if that's really the case, that 1 in 4,000 men over the age of 50 years have UBA1 mutations, then that's saying that VEXAS may be, in fact, the most common or close to the most common autoinflammatory disease that we now know of. In any event, the estimated population prevalence, at least in this population, was 74 per million. The pathogenic mutations in UBA1 are actually more common than the frequency of a lot of the diseases that were initially the diagnoses of these patients, such as relapsing polychondritis. And this cohort actually expanded the clinical phenotype. This is a recurring theme, that we're finding other phenotypes that are also associated with UBA1 mutations besides the phenotypes that I've already told you about. It's an expanding universe at this point. They found cases of dermatopolymyositis and CML in their cohort. If one even wants to go bigger in terms of a cohort and looking to see if you can find UBA1 mutations in larger groups of sequenced individuals, the NIH All of Us cohort has been looked at, and this is a paper that recently came out in Arthritis and Rheumatology. Alex Bick at Vanderbilt and Peter Grayson did this work. They looked at 245,000 individuals who had undergone genomic sequencing and looked to see if they had mutations in UBA1. They found 74 out of the 245,000 who had mutations in UBA1. And it was one particular mutation, the substitution of leucine for methionine at position 41. This worked out to a frequency of about 1 in 3,300 overall. The All of Us cohort happens to be skewed towards certain populations of individuals. It's about 60-40 women to men, 84% women in this cohort. No association with any particular diagnosis. This was actually a group of individuals who probably are pre-symptomatic, and you can see in this histogram at the bottom of the slide that in fact they found individuals actually in their 20s who had UBA1 mutation, but presumably are asymptomatic. This is telling us that these mutations are there for a while before they become symptomatic. Of course, in the women have 2 X chromosomes, and UBA1 is on the pseudo-autosomal part of the X chromosome. So the women, for the most part, will not have any symptoms unless they happen to have a monosomy for the X chromosome in the myeloid cells. If you ask another question, well, amongst older men who look like they have VEXAS, if you sequence their UBA1, what percentage of them actually have UBA1 mutations? Well, this is a nice study from Yohei Kurino in Japan looking at that issue. And he had 65 patients that were referred with possible VEXAS. 28 of them he tested for UBA1 variants. And out of that 28 individuals, 10 had UBA1 variants, 18 did not. So what that's telling us is that there's probably another VEXAS, or maybe a couple more VEXASes, or who knows how many more, out there waiting to be discovered. If you're interested in what is the differential diagnosis of vacuoles in the bone marrow, I know you're just dying to know about that. Here's a list of all the different things that can cause vacuoles in the bone marrow. So VEXAS is not the only thing, but if you're looking at individuals who have high numbers of vacuoles in early erythroid and myeloid precursors, then VEXAS is the thing that's most likely to cause that. Here's a nice study that was published just recently from the French VEXAS registry, showing that you'll remember that valine mutation at position 41 is associated with increased mortality, and in fact it's associated with an increased rate of infection in these individuals. Really, at this point, in terms of the treatment of VEXAS, we don't know what's the best treatment for VEXAS. For sure, almost all of the patients that you will see who might have VEXAS are going to be on high doses of glucocorticoids, because that's the only thing that dependably will control their fevers. Certainly IL-6 inhibition in the form of, for example, tocilizumab may help with the inflammatory manifestations. JAK inhibitors, particularly ruxolitinib, may also be beneficial in these patients. But for cure, really, you're talking about needing to do an allogeneic bone marrow transplant. And if you look at the survival rates, at least over a 2-year period of time in this particular study, it's about 75%. So the problem is that you have these individuals who are older men, for the most part, who are sort of muddling along on high doses of steroids and something else, and then you offer them a treatment that could be curative, but in a couple of years could lead to their death in as many as a quarter of them. And you can imagine that this is something that not a lot of them want to do. Well, in any event, let me just see here. I can't see my screen now, but let me see. Okay, so VEXAS, the big picture, it's a paradigm shift going from the gene discovery, going from the genotype first to the phenotype first approach. Adults may develop autoinflammatory disease the same way that they develop cancers, by somatic mutations. Throw out the textbooks. And what I mean by that is just simply that, in fact, if we look at the clinical picture that these patients have, in fact, that's like the leaves of the trees. But the molecular diagnoses may be what we're really interested in. And that, of course, are the roots of the trees that we need to be looking at. So with that having been said, here's the people that contributed to all of the work. A bunch of collaborators whose names I won't mention, but here I will just show you the NIH Clinical Center where a lot of this work was done. And with that, I will call it to a close. I thank you all for your attention, and I will attempt to answer questions that you may have.
Well, thank you very much, Dan, for the wonderful talk. It was a very beautiful story, starting from the very beginning and ending with some treatment options. So we are running a bit late, but we have some questions. Maybe I should start with some questions to Seza's talk first. So the first question is about the use of methotrexate. If accumulation or lack of metabolization of adenosine is an important feature in the pathogenesis, why methotrexate, who interferes with adenosine receptor, is not considered in these patients? Are evidences that methotrexate specifically does not work in these patients? And a follow-up for this question, the person who asked the question had a good result with methotrexate in a patient later diagnosed with ADA2 deficiency when genetic testing was available.
So we use anti-TNF mainly because of Dr. Ombrello's paper, where she has shown that if you use anti-TNF, they don't develop strokes. And strokes is, very devastating and causing one of the main causes of the complications. Now, I do know that methotrexate increases extracellular adenosine, so I do understand why this question is asked. I don't have a good answer, but I might consider it if I have a patient younger than 2 years of age, where I won't be able to use TNF inhibitors. But the current practice relies on this long-term follow-up where, the patients can be saved from strokes by anti-TNF, and we don't have that same data for methotrexate. That's all I can say. I know I don't have a good answer for that.
We need more data. Well, the second question is about the types of mutations. Some mutations are more deleterious than others. Should this influence treatment decision?
No, actually not. We don't base our treatment on what mutation there is. So, sometimes the same mutation might be associated with a more hematological manifestation and sometimes more with a vasculitic inflammatory manifestation. So there's not a very clear distinction there. And no, we do not change our treatment based on the type of mutation.
So do you regularly measure the ADA2 activity?
No, we don't. Actually, we don't. I'm not sure we would expect it to come back to normal. But no, we don't measure it. Of course, I mean, after the bone marrow transplantation, it will come back to normal. I'm not sure we need to.
That was for the treatment decision, not for the follow-up of the patients. I mean, whenever you plan to a treatment, to differentiate those with very low ADA activity or with some ADA2 activity. Well, in the same line, we know that the patient.
Good question.
Yeah. So some patients
That's a
may
good question, but no.
Yeah. So some patients have more serious hematologic manifestations, and the question is about when you would consider G-CSF as no longer appropriate and start thinking of bone marrow transplantation. So what's your threshold for this disease?
Yeah, so for the ETA2 enzyme level, that was a good question, but we decide on bone marrow transplantation on clinical grounds, whether the patient has severe pancytopenia or not, or severe immunodeficiency or not. So on clinical grounds, we decide based on that. For G-CSF, you can use it when you are waiting or preparing the patient for a bone marrow transplantation, but you can't do it lifelong because this is a lifelong disease and you can't give G-CSF lifelong. So you can use it temporarily.
Thank you. And for Dan, the recent results about the screening of healthy individuals revealed unexpectedly high numbers of individuals with variations in the UBA1, but most of them are asymptomatic, and the frequency of clones are actually a bit low compared to the numbers of clones identified in patients. So do you think there is a threshold when they start to be symptomatic? The expansion of these clones are possibly the critical issue for being symptomatic. Is there any threshold?
Well, that's a great question, Ahmet, and I think that likely there is, and it's going to be a nice thing to study individuals over time who have been found to harbor UBA1 variants, especially those that we've already associated with VEXAS, and see whether or not there is some sort of a threshold. I can tell you that in the literature, the youngest patient that I'm aware of anyway, who has symptomatic VEXAS is a patient that was published from India, who I believe was 29 at the time that he presented with clinical VEXAS. So it can happen that early, but certainly I would expect, as you suggested, that probably the variant allele fraction has to go up to a certain threshold level in order to have symptomatic disease. It may be that eventually we'll be able to monitor, that we could screen for that and then monitor for levels and predict when someone would become symptomatic.
And as a last question, since we are talking about rare diseases, the VEXAS is a relatively rare condition, although it's recently described. Maybe the most rare forms of VEXAS would be female patients.
Yes.
Could you speculate about the appearance of VEXAS in females briefly?
Yes. It's an interesting question. So, of course, females do have 2 X chromosomes. UBA1 happens to be on the part of the X chromosome that is pseudo-autosomal, meaning that a woman with 2 X chromosomes will have both versions of UBA1 active. So therefore, if they have a mutation in one of them, the other can make up for it. And so for most women that have a mutation in UBA1, even if it's a proven disease-associated mutation, they're not going to have any symptoms. When they will have symptoms is if for some reason in their hematopoietic compartment, they lose their normal X chromosome. If they develop monosomal X, which can happen, over the course of the lifespan. Yeah. And so women that have been documented to have clinical VEXAS, in fact, have a mutation on the one chromosome and then they've lost the other chromosome. In terms of their clinical manifestations, it's very similar to male VEXAS. So it's not that they have some different clinical presentation.
Thank you so much. Thank you, both of you. On behalf of ERN-RITA and EUVAS, the talks were really wonderful, and I hope that was very helpful for our audience as well. So I just want to remind the audience that there will be weekly seminars on this month to increase the awareness of vasculitis. And to close the session, I will leave the stage to Sarah Monti. Sarah, do you want to say a few words to close the session?
Yeah, the talks have been very great. Thank you so much for presenting today. Again, I remind you of the Vasculitis Awareness Month, and I hope you'll be there for the next meetings. Thank you very much.
Thank you all.
Thank you.
Yes,
Bye.
thank you.
Thank you. See you.
Bye-bye.